Hi, everyone. Welcome to the Genetics Podcast. I'm excited to be here today with Dr. Veera Rajagopal, who is known on Twitter for his prolific tweet threads. He is at Dr. Veera, spell out Dr. Doctor, and he's a scientist at Regeneron, focused on drug discovery and neuroscience and psychiatry, really focused on the genetic side of precision medicine drug discovery. So first of all, Veera, thanks so much. We're going to have a special episode wrapping up 2022 and looking on to 2023. So thank you for taking the time today.
Thank you very much, Patrick, for having me on the podcast. I'm excited for today's conversation.
Great. So we are going to just give a quick intro to Veera and a little bit about his work. Then we're going to spend some time recapping 2022. Again, if you don't follow him on Twitter and his Substack, which we'll come back to a little while later, which is an email newsletter platform, he does amazing tweet breakdowns of some of the latest science and genetics. And he's put together three of the areas where there have been some major interesting pieces of science and themes emerging in 2022. So we'll go through that first. And then we've both put together two, maybe call them predictions or areas to focus on for 2023 that we'll cover off in the end of the episode. So to start off, I'd actually just love to hear, Veera, how did you get into the amazing tweet threads that you do summarizing some of these incredibly complex papers that take normal people 6 hours to 20 hours to read and digest, and you break them down into these beautiful 10-minute threads. How did that come about? Where did it start?
So I've been like on Twitter since 2015, but I was like not using that much. I think around 2019, that's when I actually finished my PhD. I started spending quite a lot of time on Twitter, and it just happened just like that I just noticed that like things sometimes that I say are resonating in people and from their feedback. So I think it's naturally reinforced my like a Twitter activity, and I started doing it on a regular basis. Yeah, so that's how it started, and then I kind of completely shifted to writing only about mostly about the papers that I read and the summary. So one thing that I often noticed in Twitter is that people tweet about all the papers. They just retweet it or just put the title. But like when you actually take some time to read it and or give them like 2 points or something, and it kind of people like it more because people also find it more useful, and particularly students. So yeah, so that I really enjoy tweeting about it. It's mostly an activity for my own self-learning. So when you start, like when you look at an article or read an abstract, it's like might look very simple. Oh, it's clear. It's I can understand it. But when you try to summarize it, or just one sentence or two sentences on your own, you write about it. Just you start to think about a lot of questions that you didn't think when you were reading. So I really found it like very useful to understand, and naturally it became more useful for the followers, for the readers as well. I'm trying to do as much as possible like before, but I've been not doing that much when I was in academia, but trying to keep up. So hopefully we'll be able to do it in the future as well.
Yeah, definitely. And you've launched a Substack as well, which is, as I mentioned before, an email newsletter. It's maybe a little bit longer form. How did you decide to do that? And is it going to be the same things you're posting on Twitter, but with a little bit more color and less structure that the tweet threads enforce you to have certain characters? Or what's your plan for that?
I mean, I always was interested in writing blogs and everything. But, I have this typical trait of novelty seeking and, not that much of delayed gratification. So, and I lack discipline. So the reason why I was like very good, have been like successful in the Twitter is that it takes only a very short amount of time most often, and I can able to keep my focus and everything and get it out, and, right. But when I want to write a long post, I have to like really follow a disciplined practice and take more time. And I tried so many attempts in the past to start blog post. I think I even lost count of how many blog websites I started, but I kept failing at it, at some point when I easily lose the interest. But recently, a lot of my friends kept telling me that you should start a Substack. Like definitely people will love it, and you should try Substack. And the recent changes in Twitter, the
Yes.
takeover and everything, people started migrating. So I was like a bit worried. Oh, like all these years of effort building up this network, I might lose. So I thought it's high time that I started. And so far I'm enjoying it. It's kind of helping me to follow the practice of self-discipline. So because it takes more time, and people are more forgiving in Twitter if you do typos and grammatical mistakes, but not so in a blog post. So that you need to be more working on it. I really enjoy it. But in terms of what I'm planning to post there, I'm still like not very sure about it. But like I think that, so it's the thing that I feel about this attention, like focus only for short time, and so it's the same thing for others as well. So the messages will reach out to people if you say one or two things at a time, or just in a few in a thread. But if you want to ask the people to go to a blog post and read something for like that can take 15 to 20 minutes, not everyone can read it. So I'm just trying to plan in a way that I continue to do what I do in the Twitter, and that's the most successful way of communicating science. But I will also spend time to do more structured posts where I put multiple papers that fall under a theme and tell in a way, in a typical storytelling way. Still trying to find out how things work out. So for three posts I've done so based on like a list of papers, one for the Thanksgiving, and also I did one for based on the Chat GPT. I just gave an IQ test and
Yes.
summarized it, so people liked it. So yeah, so for the next post is like I'm going to write a big summary of 2022 highlights. Hopefully we'll get it out and before the new year, very close to the new year.
Yeah, excellent. I think you have at least 6 major areas. We're only going to cover 3 of those. So if people want to get the other 3, they can go over to your Substack and subscribe. It's called GWAS Stories, right? GWAS
Yeah.
Stories.
So
Yeah.
yeah. So G was it just came from my like the name G was storyteller. I think Eric Foreman, right, from Pfizer. So he describes himself as G was whisperer. So
Yeah.
so I just for fun, I think I just updated my profile saying G was storyteller, and
It's
I
perfect.
didn't really plan to keep it, but it people liked it, and like so I thought, and then okay, let's keep it like that. And so then I gave the same similar name to a similar theme for the
Yeah.
Substack.
We're not going to cover Chat GPT. It wasn't on either of our 2023 genetics lists, but I can't help but ask because I'm fascinated by it. What do you think about it? You've used it and written about it. What's your sense of it?
I mean, like, I'm not a very machine learning person, so I have a lot of friends who are there. So far, the response is that they are not like as much, you know, impressed by like how I'm impressed. But I think that it's really amazing that an algorithm can converse with you like a human, and you
Yeah.
able to get so much information out of it. And people already, I think there's like hundreds of different ways you can use it. I think it's really going to be very useful for in academic setting to write, like help with writing grant application, even papers and summarizing. I'm even worried that, like ChatGPT will take over my job in the Twitter of summarizing the.
Oh no, you've given somebody that idea. I don't think it could quite. I think it's amazing. And it's only going to get better. I do think it's mainly good at synthesizing. I haven't seen. I mean, besides some like funny examples, like you ask it to write a poem in the style of Jar Jar Binks or something like that. It is always going to struggle with invention of the new frontier, right? So I think there's, but I think it's amazing. I'm with you. I'm
Yeah.
not a card carrying machine learning person, but I'm pretty impressed with what it can do.
I just even ordered a book. There was this one of the adorable uses to write children's stories. And like you can also use ChatGPT to create the appropriate prompt to create the illustration in DALL-E, right? So there was this guy who used ChatGPT to tell a small children's story, and then asked the ChatGPT to give the prompt for the DALL-E. And he published a book actually in Amazon. I think it's been successful. probably is making a lot of money out of it.
That's
I mean, but
amazing.
the moment I saw the tweet, I ordered it because it kind of marks this occasion of this, how this chat GPT, this release of this chat GPT and how the world has been using it. So I have this book. So probably like few years from now, when I look at it book, I can remember this time. So yeah, it's really great. I mean, from a layperson's perspective, I say it's like mind blowing what it can do actually.
We'll see how frequently my wife listens to these episodes. We have a 6-month-old daughter, and I am going to make her a Christmas story book for the holidays. I'm going to do that tomorrow, and we'll see if she listens to this before I give it to her. She
You should
realized how
try
last minute.
ChatGPT and
Yeah,
also
that's what I'm
Dolly.
going to do. Well, what I think I'll do is I'll write the story and then I'll feed the prompts to Dolly and see what happens. Just one more question about you before we dive into the 2022, 2023. And in your day job, when you're not telling GWAS stories, you're a drug discovery scientist at Regeneron. What do you focus on for those who aren't as embedded in the relationship between genetics and drug discovery? What is that all about? And why is genetics so important? And why are you so interested and focused on this?
Yeah, sure. So I joined Regeneron Genetic Center like in May 2021. So before that, I was in Denmark. I did a PhD in psychiatric genetics and also postdoctoral fellowship. So I studied the genetics of psychiatric disorders. But as I started to read widely, so I have a medical background. So I was trained as a physician. Yeah, and so I have a broad interest in the genetics of all the diseases and human diseases, and started reading more about it. Then I really started getting interested more in how genetics is being used in the drug discovery research. So one of my thing, like when I moved my research career from being a clinician to a full-time scientist, it's always that you have this feel that you are maybe not, you have spent so much time, like more than 10 years in the clinical practice, and then now you're not using that knowledge. So then so I found this field of drug discovery research is an excellent opportunity where I can combine my expertise in genetics, but also my expertise in medicine, to make some like discoveries, or to do research that has more translation potential. So luckily, and the time when I realized it is actually the high time when the whole field started shifting towards this area, and all the lot of pharma companies started investing in the human genetics. So that's the background. So I got this opportunity at Regeneron. I moved there. So what we are doing here, at Regeneron, so I am part of this translation genetics group, which focuses on neurological diseases and also psychiatric and ophthalmological diseases. So what we basically do is we large scale genetic association studies. We focus a lot more on the rare variants based on exome sequencing data. And one of the major goals for us is to identify naturally occurring mutations that kind of protects the disease, and to understand what is the underlying mechanisms that leads to this protective effect. And if we can try to mimic that using drug design. And a very good example is PCSK9, and we have so many other examples now. And so this is the most important goal. But also we do not just focus on that, but also other findings, also risk increasing findings as well, because sometimes it can just open up the biological understanding of the diseases. And yeah, so that is my day-to-day job, to run large-scale genetic studies, to understand diseases and look for opportunities to help with the drug discovery at the company.
Yeah, and one of the themes we're going to touch on later is the sheer growth and volume of genetics studies, but also now the transition from exomes to genomes and what that opens up. And for those who aren't aware, Regeneron Genetics Center has been one of and probably the number one company that has taken up large scale sequencing and embedded into their drug discovery process. AstraZeneca, GSK, a number of others have made very significant investments in genetics. But whenever you go to American Society of Human Genetics, the major conference every year, it's like 10% of all of the talks are somebody from Regeneron talking about the amazing genetics they're doing.
That's only recently, right? So
Yes.
I just heard from one of my friend that this year's ASHG, who is a senior scientist from a pharma side. So he was like impressed by the amount of representation of industry scientists at this series. He
Yeah.
said just few years back, perhaps 10 or even less, the people from industry, presenting any genetics for people, industry scientists are like treated like an outcast, but the tables have turned. So such a big transformation.
Yeah, it's a big shift. Maybe one we'll come back to later. Okay, so diving first into 2022, we talked about a couple of major milestones. So we're going to dive into three different areas. I'm going to let you introduce the first one, which you've titled as some mind-blowing genetics, and we'll talk through a couple examples within this theme. So I'll turn over to you to explain what you mean by this and talk through a couple of the examples.
So yeah, I was like past few days, I was just looking at all my old Twitter threads, and I do this like been doing this for like 2 years now. That at the end of the year, I just look at all the Twitter threads and try to see what are the biggest stories, and put them under some themes. Last year, I posted like few threads. It was very successful actually. So when you asked about this podcast thing, so then I started looking into it. It's also very difficult to choose as there's so many findings. So it's just probably from my personal perspective, these are the major ones, but I'm sure there are so many other things, but that I'm probably not aware of, haven't read, or I missed it. I chose like three major themes, like three studies for perhaps in each of the theme to highlight. The first three themes are, one is like called mind-blowing genetics, findings that are really like you feel like jaw-dropping findings, and then milestone achievements, and so it kind of marks the stage where we have reached something, achieved some success in the long progress of the human genetics research. And then third theme is step in the right direction, focusing on what are the research that should be happening in the way that we think is like in the right direction, by focusing on non-European ancestry based research and everything. So the first one is mind-blowing genetics. So I think like I chose three. There are so many, and the first one is, I think it's clear anyone will agree with this. That's the biggest story of this year, is this paper that came out few weeks ago in Nature that showed how Black Death, the plague, has caused a natural selection at a speed never seen before. So natural selection is like survival of the fittest. So the genes that it's only the genes that give some kind of survival benefits pass on to the generation after generation, the variants, right? So that's the underlying theme of natural selection. We learned about this in a lot of contexts, like protection against like in malaria or sickle cell variant, and also a lot of different natural selection examples we know. But those are all process that's been happening for so many hundreds of thousands of years, but we never came across a example where a natural selection happened so drastically. So here they were able to do it. It's based on ancient DNA research. So they identified ancient DNA samples spanning three periods. One is before the Black Death. So the bubonic plague epidemic, particularly in the Europe, that is in the middle of the 13th century. So that's their timeline. So they have collected samples that span before that and also after that and during that, from two places, one in London and also in Denmark. And they were able to extract the DNA and identify genetic variations and look at how the genetic variations, the allele frequency, have shifted. So they have like 200 samples, I think, and not so much, but sufficient to do some focus on a specific set of genes. They looked at the immune related genes, and they identified like few loci that had like drastically changed in allele frequency. And the top one is in chromosome 5, where there's like two genes, ERAP1 and ERAP2, that is involved in the antigen presentation process. So like the allele on the ERAP2, it's a splicing allele, and it's 30% frequency has shifted just after the plague to 70%. So it's like the Black Death has eradicated 50% of the European population, right? So such a drastic change in allele frequency. it's jaw dropping. I mean, it's never seen before. I think that's the biggest story. And the other big thing is like always this selection come with a trade-off. So the protection, the same allele that protected the individuals from the Black Death is also putting these individuals at increased risk for autoimmune diseases that we know from today's GWAS studies and everything, like rheumatoid arthritis or different kind of ulcerative colitis or a lot of autoimmune diseases. So yeah, I think it's one of the biggest stories of 2022, and people really went crazy. Like Twitter was crazy when the
it blew my mind reading it. It really did. Like you said, it was essentially carriers of the gene were significantly more likely to survive the Black Plague, right? And like you said, that has knock-on effects hundreds of years later in some of the diseases like Crohn's, ulcerative colitis that are at much. Yeah, that one, I think it's a great choice. That one blew my mind reading it, that the technology has gotten so good as well that we can do ancient DNA samples like that at a reasonable cost from 1,000 years ago.
It also fit with the Nobel Prize theme, right? So Svante Pabo, the ancient DNA. So
yeah. And now the second one on your list, I'm glad you chose it. I know a couple of the authors on this, and I'm obviously biased, but I think it's really great.
So so there's like two fascinating papers came from Matter's group. Your past supervisor, right? So your PhD supervisor. So I was like bit in the tie to choose which one, but I think this the more. I find it like more interesting, particularly for going into the molecular mechanism. Here, the authors actually looked into the de novo mutations in the Decipher cohort and also in the Genomics England cohort, and they kind of looked into this, like what are the factors that affect the germline mutation rate. So the number of de novo mutations in an individual. So this comes from the mutation in the germline, the sperm and the ovum from the parents. On average, we know like 70 to 90 mutations, new mutations arise every generation. So they looked at the distribution, and there are some few outliers. Like I think they looked at 20,000 trios, and there are 12 individuals. They have really like more than expected mutations, hypermutated genome. Some are like really crazy, like 400. I think the highest number they observed is like more than 400 mutations. And we know that the most important factor that affects this de novo mutation rate is parental age, and particularly father's age. And this explains about 90% of the variation. And but it seems that it probably just little lesser than that based on this study. But here they are looking at other causes of this variation, particularly on the outliers. And when you look at these outliers and just study why they have so many de novo mutations in the genome, they found that at least, for a couple of these individuals are born to parents who had a genetic rare, mutation in DNA repair gene, the gene that govern the very process to prevent, this kind of mutations to passing on to the next generation. Right. So a lot of these new mutations arise because of the replication error. And this one of the individual father has xeroderma pigmentosum. It's a very well-known autosomal recessive condition because of this DNA repair gene. And they also found like some of the individual's parents had chemotherapy just before the conception. And so these are like outlier, the rare causes of this de novo mutations. And it's really, if you think about it, it's really amazing. In such a small sample, they were able to precisely identify the cause of such a increased de novo mutations in that, and paint a picture of the causes of this de novo mutation. And I think like this line of research is going to pick up. There's like one preprint that came up like few days ago. They looked into the polygenic contribution of this de novo mutation rate and tried to look into the biology by using this way. Research, they're looking into the biology of the de novo mutation rate. I think like this sample size will increase given a lot of biobanks and sequencing efforts are happening. And I'm at least particularly excited about this line of research, how this is going to progress in the upcoming years.
Yeah, I think it's also a great exemplar of how useful linked genetic and clinical data is, because a lot of this would have been really difficult to tease out without knowing one of the parents had chemotherapy or the clinical xeroderma pigmentosa that you mentioned. I think it's also an interesting, just fundamental view of biology that if a the typical genome has only 50 or 75 mutations, and then you have someone who has 400. Why is that? And that has huge impacts on disease risk for their children and so on.
Yeah. So why I was like, even, like it attracted my interest is this is kind of same thing that we do day to day. We are trying to identify large effect, rare variants affecting trait. Basically, we are looking at the outlier individuals and what is pushing them to the, to the end of the frequency spectrum distribution, right? And so it's very same as here. But here the sample size is too low. If you are actually doing an XWAS or anything, you will not be able to find it. But just zooming into it and looking at an individual, so it's a different way, like this is very important because I work in the psychiatric field, neurodevelopmental field. Often we don't have enough power to identify, like statistical power to identify association. So you need to have more than, like looking at the p values or anything. You need more intuition, understanding to identify such outliers and guess the causes and things like that. So in a lot of ways it's kind of reflected on what I did, and just mutation, considering it as a phenotype itself was like, I find it very, very interesting. So
yeah, completely agree. The third one on your list is very hot off the presses, only a couple days old, right? Tell us about that one and what caught your eye.
Still the thread is running. Getting retweeted. So I just posted a thread like yesterday night. So I was actually trying to prepare for this podcast. Sometimes findings like this are so interesting that you couldn't resist just ignoring everything. It's also like my my trait, I guess. So I just put everything aside and sat for an hour and wrote the story in the Twitter, even though I had so many other things to do. I think it's definitely a fantastic study. So what's happening in this study is they have identified the first human case of monogenic form of obesity that is caused due to a mutation in a gene called ASIP. So it's an agouti signaling protein, and it is involved in this melanocortin, signaling pathway. That is the major one response. Genes in that pathways are mainly causing this monogenic forms of obesity. So the most fascinating thing about this study is like the very first knowledge of this, protein comes like 30 years ago, based on a lab animal model. It's called agouti mice. Mainly this gene is involved in determines the coloring of the skin and the fur. And so this mice had this mutation that kind of made this gene to be expressed throughout the body. So it's supposed to be expressed only in some specific tissues, mainly in the skin, where it's role. And this mutation causes this gene to express throughout the body and also in the hypothalamus, and it starts to, disturb the appetite center, and the mice gets fat, gets hyperphagia and become fat. And so I guess it's a very popular mice. It's been used for a lot of research so far, but people have been like searching for any kind of human genetic evidence for this gene for probably this 30 years, but nothing has been found. And this is the first time they are encountering the human case. And like the features are almost similar, near phenocopy of the mice. This was a girl child, who have severe obesity and red hair and, all the other complications of this obesity, diabetes and steatohepatitis and everything. And this is part of the routine clinical work. They tried to look into the genes that are known to cause monogenic forms of obesity, but they did not find any mutation. And this girl underwent a gastrectomy surgery for the, extreme obesity. And so they collected adipose tissue sample, and then they looked into the gene expression, transcriptomic profile, comparing with the control tissue. There it is. So you don't even need any kind of statistics to look at it. There's one gene that's extremely highly expressed, like several hundred folds. And what is that gene? That's the very gene that we know, causes obesity in this agouti mice. So they went back to the whole genome sequencing data and looked at the reads, and they can see there is a tandem duplication that switched the promoter of this ASIP gene with the promoter of a nearby gene, and that gene is an ubiquitous gene. It is expressed throughout the body. Now, this gene, ASIP, is under the promoter of this itch gene. So now it is getting transcribed in every cell of the girl's body, right? So throughout the body, and including the hypothalamic neurons. And so this ASAP is it's an antagonist for two melanocortin receptors, MC1R and MC4R. In the skin, it binds with the MC1R to create this secrete pure melanin to cause this red skin and also red hair. And also in the brain, it antagonizes the MC4R, and which results in a very increased appetite. And so the patient overeats and then they become obese. And so it's amazing. They went back to a childhood obesity cohort and tried to see if there are any other patients with the same mutation. And they found four patients with the mutation. The odds of looking at it, right? It's like the same mutation in four patients, and three girls, one boy. The three girls had the very similar phenotype: red hair, hyperphagia, extreme obesity. It's mind blowing, right? So these are the findings I really like in the human genetics, and there is like a happy ending to it. So there is a known FDA approved drug, MC4 agonist, that can use it to neutralize this antagonistic effect of this overexpressed gene. And so it's a great paper, and I loved it. So I guess people also loving it so far from what I see in the Twitter.
Yes, I'm seeing that it'll get triple digit retweets for sure. And just for everybody listening, we'll add the links to all these papers into the notes in the show. So if you want to go and check them out, and we'll link to Dr. Veera's Twitter so you can read the threads as well. Your second major theme, milestone achievements. Maybe you could talk through the first enormous GWAS study.
So this is the most important. The milestone achievements, I choose three things. And the first one is it's one of the biggest finding when you narrow down the field to GWAS. If you ask me what is the biggest finding in the GWAS field, then I would definitely choose this. So there was GWAS of height in a sample size of 5.4 million. And what is special about this GWAS? It's a saturated GWAS, meaning, so at this 5.4 sample, we have found everything that you can find, all the genes and the variants that you can find, for that are associated with height. So we have reached the saturation. So in the past, we always believed that, it is impossible to reach the state because it's going to be like infinite, the sample size required to completely, get all the genome-wide significant variants, the significant variants to explain the full height variation, right? So that is explained by the whole genome. So that's a remarkable moment, right? So height is a prototype variant. So the major finding here is at this sample size, they have this, like the SNP heritability, we say, it's a heritability is the variation of the phenotype explained by the SNPs, the common variants, and it is around 50%. So you can estimate it even if you don't have any significant, genetic variants using the statistical method. And the twin heritability of height is around 70 to 80%, and of this 50% SNP heritability. But so far, the genome-wide significant variants explain, only a small portion of this full SNP heritability. And this is one of the gaps that we are trying to fill in as we increase the sample size. And so in this GWAS, that gap is filled. So now we have reached completely to that. And so the other insights from this, studies, there are so many important insights. Like when you have such a large sample size, you can go back and see what are the sample sizes you require to, to get different kind of insights from the data. For example, if I want to identify genes or pathways, right, do I need 5.4 million samples? No, you don't need. So they give an estimate of different sample sizes at which you reach saturation for different kinds of results. So, for example, you reach saturation for pathways, like all the height-related pathways, to identify pathways in 250,000 samples. So you are not going to find any new pathways beyond that sample size. If you are interested in identifying all the height-related genes, particularly that cause, Mendelian forms of height problems, then, at 1.2 million size, 2 million, you reach the saturation. Beyond that, you're not going to find. And if it's the same, but when it comes to the variants, individual variants, you reach a 5.4 million samples. And another important thing is this variants, like they identify 14,000 variants in total. They are not distributed throughout the genome as a lot of people expected under this omnigenic hypothesis, but they're distributed only in 21% of the genome. So when we talk about polygenic trait, we think that genes throughout the genome, everything at some point in the future, you will be like finding every gene,
Interesting.
every variant in the genome to show a significant p-value. And that is one of the common criticism, that this is going. So at some point, you will end up finding all the gene, but that's not the case. So here they clearly show that it's only the part of the genome, and like you start to find more and more variants only in the gene that is like more related to the height. So they have this kind of cool plot where they show different variants associated, with height at the same locus. And there's one specific locus called near a gene ACAN, which is one of the most important locus where 25 independent loci is identified. I think this is the moment like people were trying to, accomplish since like past probably 10, 15 years. And it's a huge success and motivation for the field. And so, yeah, definitely it's one of the important achievement milestone in the 2022.
I feel like I need to go back and reread it because, to be honest, when I first saw this, I thought, oh, it's another very large height GWAS. And I think probably a lot of people.
Exactly. Yeah. So a lot of people thought that it's not like there are so many million samples GWAS has come this year, but nothing like this. This is very important. This marks a milestone in our
Right.
GWAS journey.
So I guess this is almost the end to a quest in some ways, because one of the big criticisms of genome-wide association studies in general is like, where does it end? We're just going to sequence more and more people. But this is sort of showing at a certain point, you've found almost everything there is to be found, and you can still start to dive into other biological questions and so on.
I got to mention one important thing. I think it's very important to mention that. So here, still, there is one more thing to be, accomplished. It's like, even though you have reached the complete saturation, still it is only in the European sample, right? So it just doesn't apply to the non-European sample, particularly for, using the variants to predict the trait, the polygenic score. So polygenic score explains only, a part of this variance in the non-European sample. And this is because you don't have that many training samples from the other populations. So definitely we need to, so this kind of more emphasizes the importance of studying non-European population. Yeah. So that is one of the things. So when I say we have found everything we have found, so it's one applied to European sample. I want to emphasize that here. So
yeah, I think that's very important to emphasize. And also, I think the point you made stands, which is that this is about showing a general framework for knowing when you've reached saturation in any population or disease, right? And then you could apply that thinking or approach anywhere. Yeah. So next on your list is about the UK Biobank, which is an evergreen source of new and exciting tweet threads for you.
The other biggest story is the whole genome sequencing. So it kind of overlaps with last year and this year. I think the preprint was like last year and the paper published this year, I guess. So here, so the biggest story of last year was the exome sequencing. So when paper published from Regeneron and also from other companies like AstraZeneca, that, the first time we were able to look at the rare variant association at scale, like 450,000 individuals. And so this year we are going beyond the exomes and we are looking at the full genomes. And there was always this question of, people always focus on exomes, and that's the major focus for all the companies. So what is the benefit of looking beyond the exome? So what we are going to learn new that we haven't learned through it? Exomes, I think, so there was this paper from the Decode flagship paper, the main paper, like reporting about this data. I think it's a great paper. They show a lot of interesting findings that probably you will be able to find only using whole genomes. So the very first thing is, when we say exomes, it seems it's not truly like when we do exome sequencing, we are not really capturing all the, region of the coding region of the genome. There are like a lot of things that we are missing. One of the things is like the regions of the coding regions that are transcribed but not translated. The untranslated regions, 3', they have very important post-transcriptional regulatory roles. And so they identified a lot of associations in that. So then other thing is you can find structural variants. So it's very difficult to identify them using exome sequencing. So that is one of the excitation, like for the structural variants. I think they have like a lot of beautiful examples for that. and so many other insights, and also imputation panel. So you have a very high resolution of the variants, and so they have built imputation panel using which you can accurately impute variants, common variants from the genotyping array data. And here, particularly, the decode scientists are really the experts in this area. So they are the one who first actually successfully employed this imputation approach, so to identify variants all the way to down to very rare allele frequency in Icelandic population, because they have this unique genetic structure. So they were able to do that. So they have only whole genome sequenced part of the population use that they kind of apply to that. So they are an expert in this, and they have really showed off their skills in the UK Biobank. And yeah, so many other great findings. But the real question is, we will come back to this when we talk about what we can expect in the 2023, like how this data is going to be used and what are the challenges. We'll come back to it later.
Yeah, and I think it's probably fair to say most of the biobanks, well, I don't know. I'll ask this too, because I think people have different opinions on this. I hear many people say, actually, I'm going to keep doing exomes for a very long time because they cost, they don't cost very much at scale still compared to genomes, but you don't know what you don't know. Do you have an opinion on whether everyone's going to go the direction of UK Biobank and it's going to be genomes all the way? Or
yeah, I think the jury is out. So I remember the first time the discussion happened in 2000, like previous year ASHG, they were first time announcing about the whole genomes. And there was this one presentation from a scientist, I forgot, is from industry scientist who gave an initial look into the what to expect. And they did a genetic associations with all the traits possible in the UK Biobank, and they found so many associations. Then after that, when they tried to look into what are the associations that are independent of known GWAS loci, they did this conditional analysis to remove all the variants that we already know through the GWAS. Almost everything is gone. So there's very few is left. So that's one of the thing, right? So we have been doing GWAS for a long time, and what is whole genome going to bring beyond what we have learned using genotyping array and imputation? I think it's still not clear. I think one of the challenges there would be to what are the other regulatory annotations that we'll have to interpret them. So discuss a little more on this when we look at the 2023.
Yeah, definitely. The next one on your list is another UK Biobank, which I am definitely with you on this, proteomics at scale and Olink in particular. Maybe you could talk a little bit more about that.
Yeah, so I think the other biggest, it's always, I think most of the biggest stories always stem from UK Biobank. They are the pioneers in all of this. So it has been a huge success with UK Biobank industry collaboration in terms of whole exome sequencing and also whole genome sequencing, right? So, and the next biggest data, at least, from the genomic side, I think it's this proteomics that is based on a collaboration between UK Biobank and so many former partners. And they have generated a data of proteomics protein expression in the blood for 1,500 proteins at the first phase for 50,000 participants. So that is like a very big data set. So we usually, when we interpret, genome-wide association loci, one of the very common thing we do is go back to and look at whether if this variant affect the gene expression. We look at the GTEx database as a database to see the EQTLs. But often people say it's more important to also look into the protein, because probably things that you look at happening at the mRNA, but probably will not be reflected in the protein. But we never had a large database, and people started building it. The largest one was from Decode, I think 35,000. That was published a year ago. It's a fantastic paper, and now we are having 50,000 from the UK Biobank. If you combine Decode and UK Biobank, it's 80,000. That's extremely big data set, and so many possibilities and research can stem out of this data. So we had this preprint probably published in the next year. So they did some preliminary analysis to see what are the things you can do with this kind of data set. So the very first thing is, so protein represents a molecular trait that is very close to the DNA. So now you really have extremely many folds more power to identify these genetic associations. And so this can help in refining your Mendelian randomization analysis to even for drug targets or epidemiological questions. And you can use this variant associations with the protein to identify the pathogenicity of the variant. So you might be familiar with this massively parallel reporter assay, right? So where they use CRISPR to edit every possible bases in a gene and make all this possible variants and look at the some gene expression or the cell culture multiplication, like the survival assay, etc. So this is in a way kind of an MPRA, except that we have already the fully saturated, right? So list of all the lot of variants, and here the readouts is the protein. So it's very similar to MPRA, but it's scaled for like we are having for 50,000, right? So this is going to really refine all our GWAS findings and findings. And particularly, I think Decode has like a very special attachment to PQTLs and proteomics. So if you look at Carrie's talk, I've been listening for like four or five of Carrie's talk, and every time he goes somewhere, somehow goes into this proteomics data, multiomics data. And recently also, Carrie gave a talk, fantastic talk in the ASHG, the Drift Symposium that Regeneron organized. And he talked about one of the special use of this proteomics is to risk prediction, proteomics risk score. And so he said this kind of captures a different dimension than what genetic risk score. So we know polygenic risk score to identify disease risk or disease progression, and proteomics risk score kind of is not same as genetic risk score. It captures a different dimension, and probably these two will complement well. I think a lot of people are working on it. So there is like endless possibilities, and we will be like seeing so many papers in next year using this data, at least first from the industry scientists, I think.
Yeah, I think to me, what's interesting about it as well is how just the sheer complexity of the number of time points and disease states and things like that you could test. Genetics is special in that you only need to do it once, but proteomics is special in that it tells you what's happening at a moment in time in a particular cell. And so they're two very different ends of the spectrum. You only need to genome sequence people once, but you could do proteomics every second of the day if you wanted to. And it probably wouldn't tell you anything at that resolution, but you could do it every day and in every different tissue, and you'd probably learn a lot that way. So how we're going to wrestle with this question of prioritizing how we spend our scarce resources to build these kind of resources is an interesting one.
One downside is that, like we are looking at the blood proteins, and so like it's useful for a lot of secreted proteins and, like diseases related, metabolic diseases. But when it comes to, disease domains that I'm interested in, like neurological diseases, psychiatric diseases, so still, we have looked into that, it's not like clear how much this is going to be useful for that. But yeah, so I mean, apart from that, but I think it has so many potential for like other disease domains.
Yeah, definitely. I mean, this is maybe a good segue into the third bucket, because a lot of what we've talked to is centered around primarily European ancestry populations. So maybe you could talk through some of the advances here.
Yeah, I think this is even if it is the last theme, it is the most important theme. I think we should always look back into this specific theme. So I choose 3 points to highlight here. So the first one is the data set. We have the first a large-scale rare variant exome data set for a non-European population, Latin American population. And this is an effort from scientists in Mexico and also scientists in Oxford and also Regeneron, a collaboration with Regeneron. So the sequencing happened in Regeneron, but so this is like 150,000 individuals from Mexico. So I think this is one of the important milestone in the human genetics, where finally we are, we are having such large data sets for non-European population as well. And the preprint describing, the initial analysis has been posted this year, and probably it will be published sometime next year. And so this will be like people probably will use this in scientists in Mexico, and as well as other researchers will be like collaborating in Oxford and probably use it for long. And we'll be learning more about the genetics of Latin American population, and also this will reduce this, like underrepresentation of Latin American population in the human genetic studies. So some of the major findings here is one important thing that was like really amazing is that how they actually recruited these samples. So this samples like was being recruited for 20 years, since I think like 2000 around. And the scientists, they've been like visiting house to house and collecting the samples. So can you imagine they just went to 100,000 houses to collect this 150,000 sample. That is like such a huge effort, and we should, appreciate it. I think so. And it's a big effort to invest in this, right? So, so one of the major thing about this is a population or admixed population. That is, people have like different proportions of ancestry because of this admixture events between Europeans and Africans and Native Americans. And so this is a very special population, and people. So now we are starting to realize about the more use case of this population. So there are so many ways you can look. So they can improve the statistical power for a lot of variants. For example, if that is fixed in like the homologous original population. For example, if there is a variant that is present in everyone in Africa but is no one in European, right? So you will not have power either in African population or European population to do study. But if you look into admixed American population, that will be like intermediate frequency. So this gives them more power. But it also, like, when you have the same individual harboring different proportions of ancestry, you can actually remove the environmental effects and look at genetic effects. So it's always very challenging to compare genetic effects between the population, right? So if I have a genetic variant or set of variants affecting a trait, and I cannot simply compare an European with African population and just compare the genetic effects, because there are so many other factors we have to look into. But if this both the ancestry come from the same individual, right, so then basically you are like, it's like a within family kind of design, right? So you remove all the founders. So that is one of the exciting, progress in statistical methods too. I think there is a paper from Bogdan's lab from UCLA who actually looked into. So one of the problem with the PRS is that the PRS effect sizes are different across the ancestry. So what are the reasons for this different effect sizes? And you cannot tell that by just comparing individuals from different ancestry, but you can actually, look at the admixed individuals and calculate PRS only for African ancestry proportion of the genome and portion of the genome and European portion of the genome, and compare the effect sizes. You can actually capture what is the reason. So there are different ways you can use it. So I think this data set will be like a lot of great discoveries and findings will be like coming out and following years from this data set.
Yeah, that's tremendous.
Oh, that's the first one. Okay, so we have there's two more ones. So polygenic risk score. So this is getting more and more attention, and people are starting to use it, and I think already started entering clinic. But still, we haven't solved the problem of the poor portability of polygenic risk score across different ancestors. While that is being the case, then I think like there are studies that coming up to bring out other challenges that we should be aware of when we actually implement the polygenic score prediction in clinical practice. So two of this, I think it's like, I found it very important. Like one is that there is a study that was published in Medicine, I think early this year, based on the Ugandan genomic resource from the African scientists. So they show that if you construct a polygenic risk score based on European population and then predict the trait in the African population, so it's going to be poor. That we know that, right? So you can improve that by including African American samples in your training sample. So this will improve the power. But we always put the whole African ancestry in a single bucket, right? So here they show that if you construct a polygenic risk score, even using an African American training sample, the prediction can differ substantially between different African subcontinent ancestries. So they looked into the two set of individuals, one from the Ugandan region and the other from the Zulu region. And they used the polygenic risk score for, I think, LDL cholesterol. And so the polygenic risk score trained based on African American training sample explained like 8% of variation in the Zulu population, but 0.02% in Cameroon population. Such a big difference, imagine. So it's great that you're able to show it and, highlight. When we talk about portability, we also have to go and look into the fine scale ancestry of within, we cannot just use African population as a single bucket and try to use that as a category to study the portability issues in polygenic risk score. In very much in line with that, I think there is another paper that came out that showed there is also the same issue with admixed ancestry. So coming back to our previous point where we're talking about MCPs. So when we talk about portability issues, we always talk about this continental ancestry, European population, African population, South Asian population. There is this population of admixed in ancestry, right? So when you look into the polygenic prediction in the admixed individuals, even among them, if you separate them based on the proportion of their ancestry, if you have a polygenic score trained based on a European sample, it performs well only in individuals who have relatively more European ancestry in their genome than individuals who have relatively more African ancestry in the genome. And this is also an important issue we need to address. How are we going to make sure that the polygenic score will perform well in admixed ancestries, right? So we always focus on this like homogeneous ancestries. And I think it's important. So as much as we have studies that shows discoveries, I think it's also important that the studies that actually bring out these challenges for other scientists so that they are aware of it, so they can try to work out solutions for it. And yeah, so that's the second one. And the third one, I think it's probably a bit biased because I'm from India, and I'm always like, one of my all-time concern is that the poor representation of the Indian population, also South Asian population, the studies. So any studies that come out of India is like a bit more, excites me more. And so there was one preprint that came out that showed the genetics of type 2 diabetes in the Indian population in comparison to European population. So my dad have type 2 diabetes. My mom have. So my dad is diabetic for 30 years. And, my mom also is diabetic for 15 years. I know I'm going to be a diabetic very soon. So because it's a genetic risk. And so being spent a clinical practice, as a clinician in India, I know the high prevalence of type 2 diabetes. And I have spent so much time during when I was working as a duty doctor, treating patients with diabetic foot ulcer and everything. Yeah. So it's so common. And, we have some studies, but people haven't looked into this differences in the genetics between Indian population and the population. I think this is the first time they're looking at it. So one of the main, in finding when it comes to diabetes in India is that has a very early age of onset. So people get starting to get diabetes more younger age, like less than 40 years. So, and this is not the case in the European population. And what is the underlying genetics? They show that the heritability of the age of onset of type 2 diabetes is more in Indian population than in the European population. And one other interesting thing is here, the derived a polygenic score using Indian population, South Asian population as the training sample. And they show it predicts very poorly in the European population, but it predicts very well in the Indian population. We always hear about how European population-based polygenic score is performing poorly in other populations. So it's refreshing to hear it for one.
Reverse is also true. Yeah.
So the reverse is also true, right? Yes. So it's very important.
Why do you think that? Did the authors get into why the heritability is greater? I don't know as much about the genetic ancestry of India as I probably should, but I would imagine it's also a highly admixed population as well. And you're not talking about one population, right?
Yeah, that's correct. Yeah. We have North Indian population and South Indian population. So you can categorize them more as you go more deep into this ancestry structure. I think like dietary factor is one of the important, and there's definitely evolutionary aspects to it. And and one other problem here is that, they looked into the sample and, even phenotypically, they had a very big change. So average age of onset in the samples they studied from India is 40 years, but it was 60 years in the European sample. So this slightly due to probably, selection, ascertainment bias, but probably it is also the real difference. If you randomly sample individuals from South India or from India and randomly sample from Europe, people with type 2 diabetes, and look at the mean difference in the age of onset, probably it will be more, higher in India, because we know this from epidemiological studies. And probably this is because people are having variants that causing, more stronger effects on the, has stronger genetic risk for type 2 diabetes. And we are just starting to, see what is that. I mean, in the study, they identified the same loci. I don't know if you're aware of TCF7L2 is one of the strongest type 2 diabetes risk locus identified. very early in the GWAS timeline. And it's one of the most replicated finding. And they identified the same thing. But they also see that there is like variance in the same locus, but seen only in Indian population, but not in the European population. So, right. So even with the same gene that has the similar biology in Europe as well as in India, you have this, there are variants that probably you will see only in Indian population that is like having more, contributing more to the variation that discussed in India. And this is one of the problem with the polygenic risk score prediction, right? So our knowledge of the risk variants that are important to predict a trait come from the samples that we did GWAS, which is in the Europe. So we naturally miss some of the variants that are more important in the non-European context, and we don't give enough weight to those variants, right? Yeah. So that's again hitting the same problem here. So that's why it's important that we have to go and look at the genetic architecture in other population as well. So
yes. Well, thank you. This will wrap up our 2022 recap. We've gone through 3 themes, 9 papers. I'm going to talk for a minute and give you a break because I've had you talking the whole time, and let you just take a quick breather before we head into 2023. And I think we'll revisit a couple of these themes, but we both sat down and each highlighted 2 areas where we think it's worth watching, paying attention. There's something interesting going on. So I'm going to go first with 2 of mine, and we're going to flip the script. So Veera, you can ask me any questions that you want. And if I don't know, I'll say so, and we can leave the audience to follow up with somebody who really does know. My first one that I'm really excited about is the rise of whole genome sequencing in newborn screening. This, as many people may know, there is a form of universal genetic testing in many countries using a heel prick test that tests for a very small number of genes that have very clear clinical benefit, that if you pick it up early, you can do something about it. What we're talking about here is a step beyond that, which is to do whole genome sequencing on newborns. And with most of the programs, the focus is still on clinically actionable genes, so that if you find something, you can do something really important for the newborn, put them on a preventive therapy for a rare disease or something like that. Some programs, I think, are thinking a little bit longer term, like what might you do with this in a predictive and preventive healthcare context. But why I think it's so exciting and important is there's been a couple programs that have gotten really significant funding now to do this at a very large scale. So in the UK, for example, there's been about 200 million pounds, which is about $250 million for whole genome sequencing in newborns. And why I think this is so important is that we touched on this earlier, but DNA sequencing is a very special form of the medical test where you only have to do it once because your DNA doesn't change if we're talking about the germline. And so there's definitely a path towards a future where everyone is sequenced as a newborn. And that basically folds into our healthcare record and decision-making system in some way, so that the data can be used not just for early detection of very severe and rare diseases, but also towards some of the things we've just been discussing, like polygenic risk scores and prediction and prevention of disease. So I think these kind of programs are also going to drive the costs further and further down. And in a way, it's going to kind of make genetics disappear into the background a little bit, where eventually everybody will be sequenced and it maybe won't be so special anymore. But I think it's going to push us towards this personalized medicine or precision medicine, call it whichever you'd like, future where by default everybody has the option to have their healthcare be genetically guided, which I think is really exciting, versus today where it's, as we've talked about, pretty spread out. Whether you get genetic testing or not is really highly dependent on geography and disease state and so on. So what do you think about that, and what questions do you have on that?
So I'm not very familiar with this initiative, but I was just wondering if this is like a test will be given to everyone who is born, or is just in a hospital setting. What is the case actually? So
yeah, it's a really good question. So this is a pilot that it's being run by Genomics England and the NHS, and they've been working on it for a really long time. Actually, we interviewed the chair of Genomics England on one of the previous podcast episodes, and she gave a really clear overview of what's in scope and what's out of scope. And they've been doing a lot of work around making sure the questions and concerns of parents and other stakeholders are heard. What's really different about this is I believe they do intend to sequence all comers. So it will not be just critically ill infants, for example, which is a number of programs that do rapid whole genomes for
Yeah.
critically ill, which of course totally makes sense. But I think the problem that this is solving is those cases where if you'd known what the genetics was before the child becomes critically ill, then it saves everybody a huge amount of heartache and concern. So the flip side of that is you're going to test a lot of people who the result comes back negative, in the sense that it's a very positive experience where you've been told we've tested you and haven't found anything. But a small fraction of babies or parents are going to get results that say, hopefully we found something that if we hadn't found it would become a disaster in a couple of weeks or months or years.
Yeah. So, I think it's been shown that whole genome sequencing has a great value in a hospital setting, particularly, to identify diagnosis or to rule out diagnosis. That's also an important case. And I think even tweeted about a clinical trial experiment they did in a pediatric ICU unit where they clearly found a great value of saving cost and leading to a timely treatment intervention. But when you come to a population level, right? So what we do with this inborn errors of metabolism screening using youth records, and that's being done for every child born, like in Scandinavian countries and also other countries, like we have this big biobank using whole genome sequencing in that setting. And I think it's still probably that I'm not really sure like how many of the parents want to learn about the genetic risk of a perfectly healthy child. So let me ask you, if the hospital offered a whole genome sequencing for your kid, you would have accepted it? You would want to do it, or given that there is no concern, no issue with the health of your kid? So
I personally would,
okay.
but I'm 100% with you that I actually think one of the interesting things that's going to be learned through this program is who does opt in, who does opt out, and why.
Yeah.
Because there's plenty of good reasons not to as well. I personally would, but I think there's a lot of arguments not to.
Yeah. And I think the other problem, I don't know, maybe one of the common things people bring up when you talk about this, offering whole genome sequencing as like a general health checkup, like doing it. If this information, how this is like problematic in terms of insurance, right? If this information if the insurance company have information on this genetic risk and things like that, so then that's going to change the scenario, right? If they know beforehand what is the issues that that the child will have in the future and things like that. So
and
how are
this,
they going to solve about it? So
I'll use this as one way to segue into my second point as well, because I think it's really relevant here. And in the UK, the protections are a lot stronger than in the US. They're not perfect though. So for example, I'm not an expert on this, but my understanding is that you still need to disclose information about Huntington's disease risk, for example, to insurers. So it's not that in the UK, insurers can't do anything. And in the US, as many people know, life insurance is able to ask about genetic risk, but health insurance, my understanding, is typically not. But so there's no free ticket, but the UK protections are a little bit better. I don't know with the particular program how they're planning to handle it, but I think this is going to be a really key aspect of the upfront counseling and discussion with parents of what are the risks of taking part. One of those is finding out something that is going to massively impact it. And I'll give you a chance to respond to this, but the next topic that I'm going to talk about is pre-symptomatic treatment for very significant and progressive genetic diseases like ALS or Alzheimer's. And I think this is equally important there, where if you have a family member if your family member has ALS, a typically rapidly progressive neurodegenerative disease, and they get genetically tested and they have a genetic form, and then you're deciding whether you want to get genetically tested to see if you carry it, you have the same major question to consider of do you want to know, and under what circumstances? And also in the US in particular, what are the implications of getting a test that says you have a very high likelihood of getting a very significant disease, and you haven't had a chance to take out life insurance or other?
Yeah, so it's very exciting initiative. So it's just when we evaluate the value of whole genome sequencing in a clinical setting, so the study that I tweeted, like a couple of days ago, they look into how much money you save, how much of the disease burden you reduce, so it's like you have this kind of metrics to evaluate the outcome, right? So in terms of economy and also in terms of disease burden and everything. I am not sure how. I mean, I would really love to know what will be the impact of this. I think we can only know if we
Yes.
implement it. So definitely that's one of the things to see, and how it's like impacting the country's economy and thing in a large scale. So
Yeah. And you're so right about the health economics. I think broadly, my prediction of it would be 95+% of parents won't have anything. I mean, I'll even say probably 99% won't have anything actionable, and their cost will be a couple thousand pounds to the healthcare system all in for sequencing, counseling, processing. And then there's going to be 1% where the testing is hopefully going to help avoid something catastrophic. And what I don't know is how much does
mm,
that 1% cost? Somebody will be working hard to run these numbers, I think, to figure out, do we want to do this at scale? One of the, I guess, pet peeves that I have with these systems in general is that the health economic assessments often don't encompass some of the non-economic impacts. Like, what does saving 1 in 100 families from having or 1 in 500, or whatever it ends up being, from having a traumatic first 6 months of life. But that's also hard to put a
yeah,
number on, but we have to, because the health system has to weigh it up against something like genome sequencing in 50-year-olds to do cardiac PRS or something like that, and compare the cost benefit.
yeah. So I think it's great. So it's really interesting to see how this pan out in the future. So
Yeah. And my second one, which actually related to that last point, is we've seen a couple drug approvals this year in ALS, in Alzheimer's. They don't appear to be silver bullets, and there's a lot of discussion around how well they work and side effects and so on. But one of the things that has seemed to emerge from both the trial and pre-clinical data is that early treatment in neurodegenerative disease with some of these recently approved compounds may be more effective than treating people who are already very advanced with disease. And why I think this is potentially really interesting is for kind of obvious reasons, if we can intervene before we get into severe neurodegeneration, then that's a great thing. But it also opens some really challenging questions about, like the one we just covered. If you have a family member who has Alzheimer's and it's ApoE4, or a family member who has ALS and it's SOD1, or one of the other genetic variants, and you're going through the discussion of A, whether you get tested, but B, if you are tested and you're 25 years old, how do we design trials that test this hypothesis that administering a drug to an otherwise healthy but
yeah,
pre-symptomatic population might work? And I am excited for that, because if it works, it'll be amazing, but it's also it's a big challenge to think through.
yeah, I think definitely. So there's a lot of hype about the reason for the Alzheimer's, right? So I just remember writing the one of the Nature News and Views about Alzheimer's thing. So one of the challenge, I guess, like people feel that the reason why all these drugs targeting the amyloid beta pathway is failing is that we are doing the trials at a very late stage, right? So we need to start early, but also like define early
Yeah.
stage. It's like very difficult, right? So we don't have proper biomarkers or that kind of accurately tell you, okay, this is the critical point, you have to make some intervention before this. We don't know that. And to evaluate the improvements, we don't have any good way to evaluate how is the drug is working, apart from measuring the beta amyloid plaques. But we know that doesn't correlate well with the like the disease outcomes. And yeah, so many challenges. I think like one of the solution they offer is like what you said. So we need to try the trials with early onset cases. So either you can do it in two ways. One is just focus on monogenic conditions, or caused by known mutation like AP mutation, or and identify carriers of this mutation and follow up them from the very early stage, early age,
Yes.
and enroll them in the trials. And again, there is a problem with putting half of them in the placebo thing, right? So it's not fair, right? So they have this kind of a design where we switch over the groups at some point, and then yeah. So I think they're trying out a lot of different ways. And so one is monogenic condition, the other one is like clinically identifying early onset cases, like even before cognitive impairment, using PET scan to identify the amyloid plaques and earlier signs, and then enrolling them. So yeah, definitely, I think that's the next thing to do. And yeah, it's really, if it works, then that's really great. But
Yeah.
It's just, a lot of risks are involved in that. And you have to think about the patients. And
Yes. Yeah, and maybe this is a good segue to one of yours, which is the coming back to the proteomics, because it's something you just said. I think genetics is an very imperfect predictor of something like Alzheimer's disease risk, but it may be that getting rich proteomic data could help to close the loop to find a protein-based risk score, for example, that is a lot more predictive of onset than the genetics ApoE4 might tell you you're more likely to get it eventually, but something that's nearer to a real-time biomarker, I think, is absolutely what we need. So maybe you can talk about that and what you're excited about for 2023.
Yeah, I think you just reminded me of one of the most, interesting use case for this. I think the compelling finding comes from Decode paper, who they published on 35,000 base PQTL paper. So one of the way you can use it to use the proteomics data is to identify biomarkers that can inform about disease progression, particularly when you are evaluating a drug, whether it can tell you if the drug worked or not. So they have they use this Mendelian randomization to identify, so that when you do association between the protein levels and disease, both you find two types of associations, right? One where the disease is like leading to the change in the protein level, and the other one is the protein, it's being causal to the disease. So we always focus on get fixated on looking at only the causal associations and ignore the other group of associations. But actually the other group of associations might be also equally important, because they might tell you, when you're trying to evaluate some drugs, that they can give you an unbiased, marker or a tool to identify. So I think they have an example for psoriasis, where it's a biomarker DFBE1, some protein in the skin, so it's clearly a marker for psoriasis disease. And it doesn't have any causal association. It's very clear, very beautiful example for this Mendelian randomization. And you can use that to, when you're trying out some drugs that are looking at the outcome, then you can use that as a marker, probably. And I think they also have example for osteoarthritis. So that's one of the exciting use cases. We will be seeing a lot of these examples in the upcoming years, where for different diseases where this can be useful. So that is like individual proteins, but you can also use a polygenic risk score like approach, combining proteomics, all the protein associations, and using machine learning models to train scores, and use that to monitor the progression. That is another area where probably might be useful in this context, like in the clinical trials context.
Yeah, completely agree. Wrap us up here, and I think this will be probably one of our longest episodes yet, which is good because we're taking a little break over the holiday period, so people can chop this up into 2 or 3 and listen to it as they're working off their holiday meals. Yeah, so we just touched a little bit on proteomics. Maybe we can start with that one, and Olink in particular.
So like I mentioned to you before, so when we think about what to look forward in the future, so I kind of can divide into 2. One is that the 2023, what I would expect to come out in 2023, what I would look forward, and then what I would be looking forward in a little bit longer scale, like probably 5 years, 10 years. So because 1 year is nothing. So most of the studies that I would expect to come in 2023 are already in the preprint in the bioarchive this year. So if we focus on 2023, I think 2 things that I'm more excited. One is in what are the ways people are going to use the whole genome sequencing data from the UK Biobank. And the other one is in how people are going to use the proteomics data. So I think the whole genome sequence data has already been made available for everyone, all the researchers. So there are so many use cases, people are going to use it to interpret their GWAS associations. They might be able to have better find resolution at some of the GWAS locus. So one of the important challenge in interpreting GWAS studies to identify the causal genes at the GWAS locus. So the whole genome sequencing resolution might be able to help with that. And then to identify, I think people are more excited about using whole genome sequencing is to finally to go and look into the non-coding variant associations, right? So we mainly focus on the coding variant associations. And so we know rare variants have very large effect sizes. There is also non-coding variants which can have very large effect sizes, particularly non-coding variants in the regulated regions. I think the decode paper beautifully show it, right? So we always know the exons are highly, like under constraint, right? So you see less number of mutations than what you would expect. But this is also true for some of the non-coding regions, and this level of constraints are sometimes even higher than what you see for exons. So that means what happens when you have a mutation in this region where you don't see any mutation at all, right? So probably it will have a very big effect. And we are just scratching the surface of such associations. And that is one of the most exciting, discoveries we will see. But one of the challenges is that, we will be like more restricted to traditional annotations like promoter, enhancer, around the gene region. But we also need more fine annotations like tissue specific annotation, developmental stage specific annotation to know where to look at the whole genome sequence. And that is one of the challenge. And so I guess like people probably will be probably already working on it, because I always say this one of the big idea for if you're trying to start a company or anything. If you want to make use of all the UK Biobank data, but also you have something in-house that can complement it, one exciting area is to build resources for this kind of like regulatory annotations and things like that. It will be like extremely useful to capture a lot of low-hanging fruits using this like 500,000 people whole genome sequence is coming up in these years. So that is one other area. And polygenic risk score, and then laws of evolutionary based analysis, and Decode has created a beautiful imputation panel. And this is going to be very useful for a lot of non-Indian, non-European population. So they have built a imputation panel for South Asian population. I think this is one of the first large scale imputation panel that has even been made. So GWAS studies from all these areas probably will make use of this, UK Biobank whole genome sequencing imputation panel. Just yesterday, I was tweeting about paper that showed how to, use low coverage whole genome sequencing data, combine this with UK Biobank imputation reference panel to get the best imputation performance. So that is one other use case. So there is like so many things we will see how people, it's very, as I said, I'm very excited about this area for the next year, or also like next few years.
We have UK Biobank and all of the industry funders of the sequencing to thank for this great resource.
I think it's successful formula, right? I mean, there's always this gap between academic research and industry research. It is always these successful collaborations kind of show that it's possible to have this collaboration with still industry making their profits, getting out of their investment, but at the same time it's being incredibly useful for the community, academic research community, and the society. So I think it's a great thing, great examples, all these collaborations, big data sets.
And maybe you could talk a little bit about Olink and proteomics. I only really heard of Olink for the first time maybe a year or two ago, but it seems like a really interesting approach. Maybe you could explain how which I suspect you do because you know a lot about a lot of things, how that method for proteomics works compared to things like mass spec and others, and then what's different about that compared to some of the other tools that we've had available in the past.
Yeah, so you mean the way they measure the protein? I think this Olink is based on a antibody-based capture technology. So there's two very, popular ways of measuring the proteins. One is aptamers based, where, we have this short chain of oligonucleotides. They identify this oligonucleotides based on a huge, massive library where they identify the oligonucleotides with specificity towards proteins. Then you use this to capture the protein in the blood, and then you measure this using the regular DNA array technology. I think the Decode use the aptamer base to measure. But in the UK Biobank, Olink is based on an antibody-based capture, but where they call this assay, I think, proximal ligation assay, extension assay. So they have this short nucleotide chain attached to the antibody. And I think they have two antibodies that are like targeting a single protein. So one of the problem with using antibodies to measure protein is cross-reactivity, right? So it's very difficult to have this antibody with very specific, it binds only to this one protein. So by using two antibodies, you effectively increasing the specificity. So they have this oligonucleotides that when it comes to in proximity, then they bind and, like it extends like a PCR assay. And then, you measure that based on that, using this DNA array, you can measure it. So the beauty of this is you can do it on scale, like you can have DNA barcodes and you can really scale up. That's one of the problems with the proteomics, I think, like to challenges to scale, right? So I like measure thousands and thousands of proteins in an industry scale level. So that's the technology, and still people are evaluating how accurate is this, what are the false positive findings. But so far it's been great, and it's a lot of findings replicate across cohorts, across technology. That's what the published papers have shown. So this data set is going to be, exciting. And I think at the ASHG this year, already there was like representation of this data set in at least 15 to 20 abstracts in posters and talks. There's also a talk from Chris Whelan. I think he's the main lead of this analytical work group for the proteomics in the UK Biobank conference, the Winter Symposium that happened a few weeks ago. And so there's like a lot of exciting use cases. So one, mainly in the drug target discovery, this is going to enhance our resolution of genetic associations, because every time when we find an association between a common variant in the trait or a rare variant of the trait, the natural question is that how this variant is affecting the protein, right? So is it increasing the protein or decreasing the protein? Because that is the fundamental information that we need to propose a hypothesis that this is how you are going to treat the disease, right? So, and it's going to be very helpful for that. And so looking at the association at the proteins. And so there are a lot of times, there's two kinds of genetics: reverse genetics, forward genetics. The classical genetics that we do is that we start with the phenotype, do a genetic association, we identify an association, then go back to the genome and the locus and identify the gene. And the reverse genetics can be like, you start with the gene, this gene is important, it has a specific role in this disease, and what is the consequence of mutations in this gene in the population. So using this proteomics, you can actually combine these two. For example, you first discover the gene, then go back to the gene and look at the associations at a finer level with the more powered associations with the actual protein. Then you can identify important variants. What are the missense variants that are more important in this protein or pathogenic, or are the loss of function variants? Then you go back to the population data sets and look at these associations and increase the power right? Because now you are essentially reducing the number of variants to truly affecting variants, right? So that's one exciting area. And predictions, we already talked about it. And we know, like, we have a lot of ways to predict the pathogenicity of the variants computationally, right? We have different ways to say if a missense variant is pathogenic. And so I think like this has been an excellent data set to create another, like such a prediction score. By combining with other scores, you can even improve the performance of this prediction. So you can, say if it is affecting the protein and in what ways it is affecting the protein. So that will be like very useful in a clinical genetics as well as in the population genetics, when people are imagine going back to a database like Nomad and looking at the genetic variant, and then also you're getting the information. Right. What is the effect of this variant on the protein level? Is it like increasing or decreasing, or what is the scale of this association? It's going to be amazing if you have this information in the future. And I think like a lot of other ways, Mendelian randomization, both for drug discovery as well as in the epidemiological studies. So a lot of questions, interesting questions you can answer, because now you can have more stronger genetic instruments, because like protein is very close to the DNA compared to a phenotype. That's like a very distal consequence. So essentially by using this protein, you can have like more stronger instruments for doing this causal association.
Yeah, I couldn't agree more. I think UK Biobank is the gift that keeps on giving. I just want to say thank you. This has been an amazing conversation. If I can quickly summarize, we talked about tweet threads, chat GPT. We talked about natural selection through the Black Plague and its effect on modern day, number of milestone achievements, the largest height GWAS ever, which I learned is quite a definitive moment in the field. We talked a lot about exomes, genomes, proteomics, a step in the right direction, as you put it, in some large scale, more representative populations, including Mexico, India, and Africa. And then, I think we just covered what's in store for 2023. But I'd like to say thank you for taking the time out of your holiday.
I'm really sorry for extending it for long. I think probably one of the longest in your seat.
No, this is like the Joe Rogan length we've gone for. We haven't hit the 3-hour mark. I think we're well below 2 hours, which is great. I, like you, love to talk about this stuff. So I could be here all night. So thank you. Happy holidays. And
thank you very much.
We'll do this
again soon.
Happy holidays
to you. See you. Bye.
See you soon.